Thank you. All right. So, hello, everyone. I'm Amar Kumar, PhD student here at McGill University and Mila Quebec AI Institute. And I'll be talking about one of the recent works that we published out. It's called Prism, High Resolution and Precise Counterfactual Medical Image Generation using Language-Guided Stable Diffusion. And this paper is an oral at Middle, which is medical imaging for deep learning. The conference is happening in Salt Lake City, I guess next to next week. So this is a sneak peek even before the talk is actually given at the conference. So let's jump right into it. And before we get started, I would like to thank my authors and collaborators. This work was done in collaboration with Google Research, and the other authors were Anita Mohammed and my supervisor, Tal Arbel. So a bit of background about how deep learning has been useful to us, and thanks to the other speakers who actually focused the attention on different aspects using LLMs, using it for different tasks such as classification. I guess Danny mentioned it in the beginning. So we have been using deep learning for a variety of medical images on the right and for an innumerable list of tasks. And the list is non-exhaustive over here. And these images could be mammographic mass classification or segmentation of lesions in brain, or even breast cancer metastasis detection. The problem range is huge. But the critical question lies, why haven't they been integrated into any clinical workflow? And I guess the previous speaker also highlighted this a bit, that what are the issues with them? One common issue is, although their performances might be amazing, these models are essentially black boxes, which is you throw a bunch of things in and you get some output. You don't know what's happening in between. So with that, keeping that in mind, let's try to analyze this new framework. And here's a screenshot from two of the conferences, MICI 2018 and 2023, where radiologists and clinicians have talked about this fact that AI in practice is good for medical imaging, but we don't really understand how they arrive at a particular conclusion. And essentially, there is a need to open up this black box and understand the underlying mechanism, which is important for a decision to be made. So the outline for this talk will be, I will talk a bit about the problem and motivation that is needed for this kind of setup. We'll talk about Prism. We'll go into a bit more detail about the implementation of these architecture. Next, we show the capabilities. And finally, we talk about clinical impact and how these models could be useful, and we'll conclude this talk. So in terms of medical AI reliability crisis, we have seen models that are deployed in different contexts, as I just mentioned. And some of the critical problems that we need to look at is first spurious correlations, where model associate with, instead of looking at diseases, they start looking at shortcuts. One such shortcut is they start looking at medical devices instead of pathology. There is a huge data imbalance, underrepresentation of data. So anyone who has experience working with data in medical imaging, you must realize that the field is not as luxurious as natural imaging, where you have billions of data available directly to you to train your models. And in fact, there is a huge data imbalance for specific disease, for specific demographics, and all the other criteria that you can think of. In addition to that, there is very limited annotation, because if they have to be annotated by expert clinicians and doctors, that takes a lot of time and puts pressure on the healthcare system. Finally, there exists classifiers paradox, where you need bias mitigation methods to understand biases in the data itself, and then propose methods to mitigate those biases. So essentially, we needed a model that could address some of these factors in certain ways, or at least attempt to address them. And here comes vision language models, and we have been seeing them all around. And they have been great at synthesizing images. Two of the examples, and these images, I generated them myself. using one of the tools from Stable Diffusion, and you can see that the images are pretty high quality. But can we leverage them in the context of medical imaging? That's the real question. And essentially, this foundation model proposes a new paradigm because of these three critical verticals. The first being they provide language guidance. So essentially, one can have precise control over image using text descriptions. You can do a sort of transfer learning where you can learn from a massive pre-trained data. And here, massive is not as large as the natural image. I'm talking about like 100,000 medical images. But even training on them, you can start synthesizing data that is generalizable to other fields. And given the development in technology and these foundation models that have come out, we are in a paradigm that leads to high resolution image generation that can be aligned to text. So basically, you can use text to control these high resolution images. And now comes the question that can we use these foundation models that I just showed for medical domain? The short answer is definitely yes. The long answer is how do we do that? So let's dive into a bit more detail of what are the things that we essentially need to look at in order to tailor these foundation models to medical imaging domain. And there are two key components to any foundation model, or to most of the foundation model. They are image encoders and text encoders. Here, I'm taking one particular text encoder, which is known as CLIP, trained on a contrastive loss. So that's like a bit more technical detail. If you want me to go in more detail, feel free to let me know in the chat. So these image encoders are essentially a bunch of images on which these foundation models were initially trained on, and they are diverse in multiple ranges. And at the same time, clip encoder is a huge corpus of text data on which these models are trained. And essentially, the notion or the intuition that we need to develop is somehow we need to control these two together so that we can fine-tune a medical foundation model that works on medical images. So this particular framework, Prism, that I was mentioning, is built on the backbone of Stable Diffusion 1.5, and essentially has three key components: VAE, which is variational autoencoders and decoder, a unit, and a clip text encoder. So what these components do is VAE is mostly for controlling image representation. Unit is the generative component, and clip encoder is the conditioning mechanism. So essentially, if you think about it, if we type something as natural language, and if we want to control the image, it's the bullet B, which is we need to control the generative component such that whatever conditioning we are giving through text, that can be combined with image representations. So if we look at Prism architecture, out of these three moving parts, we did a bunch of ablation, and we realized that we don't need to train everything because there is billions of parameters that needs to be controlled. However, the most effective way to train this is just train the unit part. And that's where the secret sauce for this architecture lies, is you train only the unit component, which is the generative component for medical imaging task. And no matter whatever encoder representations you get, either for image or text, that is automatically taken care of. So going into a bit more detail about the architecture, I'm sure this looks a bit bulky, but I'll try to break it down into smaller chunks. So if you look at the detailed overview in the central part that shows stable diffusion over here, the first components, which are the VAE encoder and decoder. And as I mentioned, that these encoders represent how image is going into, or image is transformed into a latent space, or from latent space into image-based representation. So this remains frozen. We don't train it at all, which means that the initial stable diffusion, which is trained on natural images, it remains as it is. The second component, which is the clip text encoder, which essentially deals with language, even that remains frozen. So it has no clue about the medical context or annotations directly. However, the central part, which is the denoising unit, that is what is trained in order to fine-tune stable diffusion so that it can be generalizable for medical imaging tasks. So given this entire structure, the only part that trains is this central unit, which takes in image encoded representation and also takes in clip encoded representations and merge those two through cross attention blocks, which is essentially the standard way of combining text and image representations. So now if we look at the overall architecture, what is this framework trying to do? As a clinician or as a researcher, you can pass in the image. I guess my cursor is visible. So you can pass in an original image and you can pass in a text. So here I'm passing an image which is defined as I original. And my text is chest X-ray of a patient with lots of medical devices. And as you can see that there is a medical device as pacemaker. And if you zoom in, there are other devices such as tubes inside this radiology. What's happening is the user put in an additional edit text, which is chest X-ray of the patient without medical device. So the only difference from the original text is that you change with lots of to without. And this model, what it does is in the output, you can see that this medical device and others are removed. Now that is the holy grail for this entire architecture. We are not providing any annotations. We are not providing where these medical devices are located, and we are expecting the model to semantically understand that what it should do in order to edit this particular image. So there is another artifact which I'll talk about, an inverted image in some time, but ignoring the inverted image, this is the output that the model is designed to take. And all that is happening with this backbone of a foundation model, which is trained on natural image, this stable diffusion. And we fine tune just this central component, the unit, for medical imaging task. And it's very beautifully able to perform these tasks without any extra supervision. So if we look at this task of language-guided image editing, it goes through several steps, and these steps can be divided into three main components and can be summarized by the phrase that you see over here. We are combining DDIM, which is denoising diffusion image inversion, with null text inversions for precise identity editing. What's happening here is in step one of this editing, we are performing image inversion. So essentially what that means is you take the image, pass it through the image encoder, and you land up in the latent space representation. What if I don't want to edit anything? I just want to check if my latent representations are good enough to capture every detail in the original image itself. So essentially, if I don't want to edit at all without any text, I'm checking my image inversion. And if the image inversions are bad, essentially that means your model is not able to represent the input image to the node. Sorry, I'm just seeing some chat here. No. So this image inversion, if your model is not able to perform good inversions, that essentially means that the model does not have enough representational capacity to make sure that your images can be edited. Step two in this case is we perform language-guided editing, which is using text embedding to guide the denoising process. So this is what is happening in the central region via cross-attention, that when a user inputs a natural language, these are converted into text editing using the text encoder and then combined using cross-attention through that unit and goes through the usual denoising diffusion process. And in the end, we also use an extra layer of filter, which is known as clip similarity scoring for quality evaluation check. So even before these images are synthesized, we perform an automated quality evaluation check to make sure that whatever we are seeing is actually a good representation of what we wanted from the language. So those are the three main steps that is involved in this language-guided image editing. And a bit of math, I hope I'm not making it too complicated. So this clip score that we have is defined by this short equation, delta I times delta P upon modulus of delta I and delta P, where delta I summarizes the image editing difference and delta P summarizes the prompt editing difference. So essentially that means whenever we are synthesizing an image, we want to check what is the quality of that image. And we want to also make sure that the quality of the image is in alignment with whatever text the user is providing. So it's not like you provide a text and the image editing is doing something else. To ensure that, we have this clip similarity score, and we make sure that if this clip similarity score drops below 0.1, the samples are automatically filtered out. Essentially, that means these samples are bad samples and we want to ignore them. So once that is done, once we understand how this architecture is implemented, now comes the fun part. What is Prism's capability and what are the things that it can focus on? It can do a lot more than this, but the three main ones are it's doing a high resolution image generation. And I believe, to the best of our knowledge, there exist only three models that is able to do so. One is by Google, which is called Biomed and the other one is by Microsoft. The third one is what we are releasing over here. We can perform free-form image generation, which essentially means if I don't want to use the model for editing, I can simply use it for synthetic data generation. We can synthesize a lot of images by just putting in text, and Prism is able to generate these high-quality images. We can have selective attribute control, where if we want to preserve specific images or preserve specific properties, we can do that using Prism. We can also use it for counterfactual image synthesis, which is explainability of the image itself. Here is some examples of Prism image generation technique, and the text prompts are given at the top. So chest X-ray of a patient with no known disease with pleural effusion and support device, and cardiomegaly and no devices. What you can see over here is in case of no known disease, you can see in those yellow box that the costophrenic angle of chest X-rays are clearly preserved. However, in pleural effusion, those parts are destroyed, which is how disease is reflected. And the arrow shows that it's also adding in some support devices to make it more realistic. Analyzing some other disease like cardiomegaly, which is abnormal enlargement of heart, you can see the central region is abnormally enlarged, showing that the heart is occupying a lot more than it actually should. So the patient is definitely not healthy. So going into a bit more detail about the capabilities of Prism, it can perform device removal, which is a very difficult task given the nature of these devices. They can be present anywhere of any size, and essentially no metadata or annotation is available. It cannot be, right? A patient, when they go for a chest X-ray scan, you cannot control what devices they have, or they might have some life supporting devices, which essentially needs to be there. So the previous state of art, which was also published by us, you can see a patient with lots of medical device. And if we try to edit it using this other method that we proposed last year, which was also the shortlist for best paper, we are able to remove devices, but not until great extent. So that was SOTA before Prism, essentially state of art before Prism was released. But here is an animation, I guess you can see on your screen, that it's those tiny regions where you have these devices, and you're essentially giving the text prompt that chest X-ray of a patient with loss of medical device, change it to without any medical device, and you can see the fine edit that all these devices are clearly removed in this animation. And I'm also showing you a difference image, which clearly indicates these tiny regions. So you can see the capability that it's able to perform all those fine editing without any metadata that is needed. So Prism is capable of removing all the medical devices without any training labels for wires, pacemaker, or any device that is present there. Revisiting the problem, which I talked about initially, which was about opening up the black box. So imagine a patient comes in and you have a black box classifier, which says essentially the patient is sick, but we want to know more details. Sick, why? And previously, these problems are addressed via some of the method, which is essentially GradCam, that highlights important regions that what am I looking at, or what are the doctors looking at in order to make a decision. But as you can see that the image on the right-hand side, that is not definitely the best way to show why is the patient sick, because there is two huge blobs, which is not very interpretable for human. So this problem of opening up the black box can be addressed via counterfactual synthesis. So essentially, that means this person is sick. What would the person look like if they were healthy? So imagining a counterfactual scenario that what if this thing was not there? And essentially, the goal of a counterfactual image generation is we want our model or any method to make minimal changes, belong to the correct target class, which means that if the initial patient was sick, my synthesized image should be healthy and it should show realistic changes and not just an adversarial attack. So keeping that in mind, for explaining the black box, Prism is able to explain that very well. And here is another animation where the initial patient is with cardiomegaly, which is again abnormal enlargement of heart. And if you can see, the model was asked, what if this patient was without cardiomegaly? And you can see that Prism focuses on relevant features associated with disease pathology, and it's creating that mass in the lung that it should not be this enlarged. And you can see in the difference map that just these areas are highlighted red, and that shows Prism capability to focus on relevant feature that is associated with disease pathology. The next one is explainability, ignoring other confounders. So I just said that it can focus on disease pathology, but what if the disease pathology is difficult and there are confounders, which essentially means that what if the disease is associated with something else? Prism is very capable of ignoring those scenarios. As you can see over here, pleural effusion, which happens in this area of the lung, and there is a bunch of wires that is existing in that region, and you can see the wires are not being removed. Instead, the patient is made healthy, which is a huge addition to the part that it's able to ignore the confounders and focus on that particular understanding of the disease itself. So Prism capable of ignoring the confounders, explaining, and also editing the image. In addition to that, here are some more examples other than just the animation. So these images show that Prism can focus on the disease, and these were also edited using text prompts. And the details about these text prompts are available in the original paper, and I'll share the link at the end of the talk. The other capabilities that can be very useful for a deep learning based approach is these image editing devices. So here are some more examples, and not only removing the device, but also adding devices. And also a baseline comparison is made using a GAN-based approach, which is like generative adversarial networks. And you can see that they are not that good in showing performances, especially for removal of images. So the details are there in the original research paper. So if you are interested, please do take a look. The last question, that what do we do with these images? It looks great for explainability. It looks great for adding or removing devices, ignoring the confounders. So here is the last vertical, which is also, I feel like, one very strong area where prisms can be used, is this area of synthetic data generation. The synthetic samples that is generated from Prism can actually help improve the performance of classifiers. So as you can see, the original data, which is the real data that we have, you can see performance on different diseases. I'm just showing 4 of those diseases. These are accuracies that is reported for pleural effusion, cardiomegaly, no finding, which is essentially healthy subjects, and support devices. And if you randomly augment with samples taken from Stable Diffusion, you get minor improvement or no improvement at all. But if you synthesize samples from Prism and augment, so original data plus the Prism, you can see there is a clear jump in the performance across different diseases, especially this disease pleural effusion. Therefore, that leads us to the conclusion, Prism can synthesize images that are helpful for improvement of downstream classifiers. And this can be of great help, especially in the data regimes where we don't have a lot of real data available to us. So the fact which I was talking at the start of my talk, that what if we do not have that many medical data that is directly available to us, how do we deal with that? So Prism can actually bridge that gap where we don't have a lot of data availability. In conclusion, this VLMs in medical AI is very useful, but need to be analyzed very carefully. Prism, one of the foundation model that can be successfully adapted to address critical bias issues in medical AI. And we have shown that Prism is able to synthesize high-resolution, high-fidelity, language-guided counterfactual images. And these synthesized images can also be used for improvement in downstream tasks. Some of the future directions, so this paper was also extended to a lot other papers, and I have attached the QR code for all of these papers, where we show different applicabilities of Prism, and they are accepted or they were already accepted to CVPR workshop proceedings, two of them, and the last one was literally accepted 2 days ago to MICAI, which is showing the capabilities of how you can use reinforcement learning to actually even further improve this synthesis at high resolution. And we are also showing the generalization across different modalities. So we show it on skin cancer data set, we not only chest x-rays, but other imaging modalities where these can be useful. I guess Jimmy will share these slides with you. So if you're interested, you can scan the QR code for each of these papers and take a look. Again, I would like to thank my collaborators and co-authors for this paper. The paper is available at this particular QR code. If you're interested, please do read. And if you have any questions, feel free to reach out to me. Also, thanks to all the collaborators who contributed to this research and our research lab here at McGill University. And thank you to my audience for listening to me, and I'll be happy to take questions. That's essentially my LinkedIn. If you are interested in connecting or reaching out, feel free to reach out. Sorry for exceeding the time. I guess that is within the time limit, Jimmy, but if there is any question, I'll be happy to take them at this point.
Yes, fantastic. So we will include all the links in the follow-up email that should drop in your inbox later tomorrow, along with the YouTube playbacks. I know there was a lot of links in the presentation. We do have one question. Let me see if I can synthesize it. It came during your discussion around the black box.
Yeah.
And the question is, when removing devices, how do you deal with removing pathology also?
Yeah, so that it's a very good question, and essentially that's the problem of confounders. Those two can be associated, and in fact, in some of these data sets, they are associated together. But that's the capability of Prism because it's trained on approximately 130,000 samples. There is quite a lot of samples where it actually understands what those devices mean and what a particular disease pathology means. So it's able to delineate or disentangle those two representations. And when removing devices, it attempts to focus only on devices. And we have statistically shown in the paper that that indeed is the case that is happening. And essentially, this entire line of thought that can we disentangle these two representations using the approach of Prism, that was one of the papers that I was showing in future directions. And if you're interested, please do take a look. I guess that summarizes my answer to that particular question. But we have a whole research paper just on that topic, that how do we make sure that those two components, medical devices and disease, are separated.